Hello, I'm Doris Cesenarro, and today I'm going to talk the investigation created by me and my college that is called Effectiveness of the Extract of the Lead of Mentha arvensis mint and of the bulb of Allium sativum garlic as natural insecticide in the control of Planococcus citri white cochinilla. This investigation is divided in four parts: introduction, methodology, results, and conclusion. Introduction. Regardless of the location or crop, pests, and disease that affect a crop production in quality and yield have to be controlled. The most practical way to avoid this problem of to use an integrated plan to break the life cycle of pathogens. However, due to lack knowledge or poor practices, the use of insecticides with harmful effects of the soil and crop, decreasing the quality of yield of these with long-term recovering time. One of the biggest problems in the insects from the Aphilidea family of the suborder Sternotia, which contains aphid, white flies, and mealybugs, with field on plant matter forming harmful pests. In this sense, the current project will carry out elaboration of insecticides from the leaves Mentha arvensis and Allium sativum for the control of Planococcus citri. These pests affect auricular plants such as tubers, bulbs, roots, fruits, vegetables, etc. We will evaluate the effectiveness of the insecticides in the control of Planococcus citri to determine which has greater significance. For the method, we divided in 5 parts. First of all, we have the identification of white mealybug, as we can see in the figure 1, the pet identification. Then is the biopesticide based on Mentha arvensis. In the figure 2 is the removal of impurities Mentha and arvensis. And the third one is biopesticide based on Allium sativum. This is the figure 3 Allium sativum motor concentration. Then we have sample, as we can see in the table, 2 test coats. We divided in test coats with Mentha arvensis, Allium sativum, and in the figure 4 we can see the Codiaceum variegatum picture with coats. And this monitoring and control is the last part of the method. Daily measure were made of percentage of mortality of Planodon citri with the application of the different biopesticides generated every 24 hours for 7 days after the first application of the biopesticide. The predicted application was given every 38 hours after the first application of the pesticide. Then we have the results. The average range and standard deviation of the number Planogon citrum found that were calculated for a basic statics. First we have the table 3, that is average mortality of Planogon citri by concentration. The table mortality average shows that on the 7th day the concentration 150 grams 21 was the only one of killed all the Planococcus citri. Then the table 4 is the variance of mortality of Planococcus citri. From the table of the variance of mortality, it is observed that the 7th day the concentration MC3 no data is found because MC3A, MC3B, and MC3C managed to eliminate the Planococcus citri, in addition to slight variation in the control due to that of a specimen study in T2 on the 6th day. In the figure 5 we have the average number of days in day 7 IBM SPSS. On day 7, AC1 has now yet extended 33% removal, showing sign of clogging, while NC3 has archived 100% removal of Planococcus citri. Analysis of the variance ANOVA analysis of variance will be performed in Microsoft Excel with a significance of 0.5% and in IBM SPSS statistics 22 with no of importance of 5% and the method mentioned in chapter 13 of probability and statistics of engineering and science will be applied. At least the conclusion, natural insecticides has been developed through the process of maceration of Mentha arvensis, mint, and leaf garlic bulb in a different concentrations, being the most optimal obtained from mint, obtaining very effective results in elimination of white cochineal in each of the trials, while the insecticide of garlic obtained a low removal. The insecticide elaborated through the maceration of garlic bulb obtains a removal similar to the removal found in the thesis effect of 3 doses of extract of 2 species of garlic in the control quinoa mound larvae with obtained ampelograms and to a lesser extent A sativum being 37.31% and 26.95% respectively, a result that is similar to concentration AC1 and AC3. The insecticide generated by the leaf of Mentha and a bulb of garlic are related to those mentioned in plant extracts in the control of pests in agricultural crops, in which it is mentioned that these species are effective in the elaboration of biocides of the control of the Aphidoidea family. Thank you for listening.